to today's webinar, Chop Your Protein Production Timelines. I'm Cassie Soltman of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by MaxCite. To learn more, visit www.maxcite.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I'd like to now welcome our speakers. Caroline Romanelli, Sales Manager, Mid-Atlantic and Southeast for MaxCite. Megan Embrey, Senior Field Application Scientist, Mid-Atlantic for MaxCite. And Dr. Jim Brady, Senior Vice President, Technical Applications and Customer Support for MaxCite. Caroline, you may now begin your portion of the presentation.
Thank you so much, Cassie, and thank you all for joining us today. We're looking forward to exploring the transfection capabilities of the MaxCite with you for the application of protein production and how it could save you both time and money. Just a quick legal disclaimer here. All of the data that we are sharing with you today is either owned by or granted permission for use by MaxCite to be presenting. In terms of agenda, I'll just be doing a quick introduction to MaxCite as a company and an overview of the technology before Megan and Jim dive into the scientific content surrounding optimization and scale-up of protein production. The introduction will be fairly brief, but we are always happy to have follow-up calls to explore any questions you may have. Or please use the Q&A feature and we will address at the end of the webinar. The last slide will also have my contact information, so please feel free to save that too and reach out with any other questions that you have. We're really looking forward to the content here of the workflow, optimization of the chosen line, antibody production, bulk and mini pool strategies, cell line development, and even large-scale transfection. So getting into MaxCite's history. MaxCite is a unifying platform for cellular engineering. Over the years, we have solidified our foundation of robust instrumentation and software that is capable of delivering virtually any cargo to any cell type, and from 25 microliters through to that 1 liter of concentrated cells with truly consistent results across that entire volume range. Arguably, and my favorite asset of MaxCite is our scientific support team. We have been focused on electroporation for over 20 years. And when you are part of the MaxCite family, we are dedicated to supporting your research and manufacture with our years of expertise in this field. And our third pillar is our partnership support, both in regulatory matters, but also in technologies that are used upstream and downstream of the electroporation step. We have built and pride ourselves on what is truly a customer-centric focus for optimal results. And MaxCite has proven to be an innovative and experienced partner for over 20 years in this space. We were founded with the idea that cell therapies would be a critical need for sick patients in the future, and that a scalable manufacturing platform was required for them. With that as our guide, the first MaxCite transfection platform was launched in 2000 with an FDA master file soon to follow. And aside from cell therapy, it became clear that scalable transfection methods were valuable in the drug discovery and protein production space as well. And we now have a wide customer base in both areas of cellular engineering. We are on our third iteration of instrumentation we call our Expert platform. We are publicly traded on the NASDAQ, and last year we moved into our new headquarters in Rockville, Maryland. And that really allowed us to expand our R&D capabilities as well as manufacturing capabilities. So we are always able to supply our clients with consumables for their research. And in a few short weeks, we will be launching our Expert VLX instrument, allowing for that 1 liter input of concentrated cells for transfection. Our Expert platform is a family of 4 instruments that enables our clients to use a single platform from concept to clinic, starting with our ATX for smaller scale R&D experiments, moving to our STX, which is larger scale R&D as well, to the clinic with our GTX, which is used in the GMP environment and has that FDA master file attached to it. And the transfection efficiency for each of these instruments is exactly the same. So when you move from R&D to manufacture, there's no re-optimization time required for scaling, which does save you time and money. And all the way to the right here is our VLX which Jim will get into a little bit and is the instrument that will be launched in just a few short weeks. Our scientific team is truly the best asset that MaxCyte can offer, and we are always adding to our scientific team that have broad areas of expertise, which allows us to best support you. Looking at our field scientists, our R&D team, and our process development team, you can see there's almost 1,200 years of total experience in a broad range of applications. And they all work really collaboratively to ensure that we are keeping up with all the scientific trends, so we are in place to support any application that you are interested in. We really follow where your science goes. Oops, apologies. And this team is also globally distributed. So if you have multiple locations or have R&D in one location and perhaps work with a CDMO, academic institution, or a manufacturing plant in another part of the world, we likely have a scientist in place that can help with your team in their local area. They're always ready to help design or troubleshoot experiments, to train new users, and to truly share those tips and tricks that will help optimize your results. At a very high level, transfection using the MaxCyte is done in 4 simple steps that Megan will elaborate on in a little bit. The 4 steps are combining your concentrated cells with your cargo, loading into our consumables that we call processing assemblies, connecting to the instrument for the electroporation step, and then continuing the cell culture and analysis specific to your application. Our scientific team supports each of these steps in various ways that are all critical for successful experiments. I've mentioned virtually any cargo a few times, and here is a sampling of the most common cargoes we see, but this is not an all-inclusive list by any means. This is where you would work with your field scientists to help optimize the cargo that you are looking to deliver. And these cargoes are used to express a variety of molecules, such as difficult to express proteins, or perhaps even large quantities of easier to express proteins. Gene and base editing tools, large amounts of transiently transfected cells for cell-based assays, antibodies, bispecifics, fusion proteins, and even viruses. The system is truly agnostic cellular engineering. Apologies. Using this platform, folks are publishing monthly. So there's a plethora of data out there that may pertain to your research. And if you're interested, please ask and we can help get you to a data set that would be of interest to you. And I apologize, I did skip over this. Oh, sorry. We seem to be missing a slide in here. But that's okay, it's okay. I apologize, everyone. The challenge with a live presentation. So at this point, I'd like to turn this over to Megan and Jim to dig into the science. Megan.
Thank you very much, Caroline, for your great introduction. I did want to review our agenda again to just kind of highlight the goals of today's webinar. We really want to share case studies that highlight MaxCyte's electroporation technology that help you reduce your protein production timelines. Or in this instance, because we are specifically focusing on CHO stories here, we decided to call it how to chop your timelines. So first, we're going to focus on areas where MaxCyte allows you to rapidly optimize your electroporation and protein production to improve transient titer. We'll then shift our attentions to stable workflows and show a few strategies for stable cell line development. Finally, we're excited to share a case study for large-scale transient transfection in a closed process using our newest instrument, VLX. So looking again at the MaxCyte electroporation process, I really wanted to focus on 3 areas where MaxCyte can save you time with our simple and straightforward process. First, in step 1, we can see that we save our clients time by having one optimized buffer for all cell types. This means that MaxCyte users are not wasting their precious time and reagents by trying to find the ideal combination that will give them the best electroporation buffer for their unique cell type. Next, and this is something we'll highlight in our second case study, our consumables are seamlessly scalable, which means any condition
that you optimize in the smaller processing assembly here on the bottom in step 2 can be directly applied to the large-scale applications, which are using our processing assembly there at the top, which is 100 mil scale, without any type of re-optimization. And then finally, in step 3, really the star of the show, the MaxCyte electroporation platform. As Caroline mentioned, we have a range of powerful instruments, all with an integrated touchscreen running our intuitive expert software. But beyond this software design, MaxCyte system also provides its users with a very intuitive electroporation energy selection process. So unlike other systems, MaxCyte actually provides its users with preloaded electroporation protocols for any cell type. This means that we're removing that time-consuming step of having you design your own protocols without necessarily knowing what you're doing with electroporation. Instead, MaxCyte provides you with a highly optimized, highly tested electroporation protocol list that you can be confident was selected and developed for you to use for your cell type. Now, what we're displaying here today is not a comprehensive list, but some of the more commonly used cell lines, as well as mortalized, primary cell lines. Specifically for protein production, we have protocols for CHO, HEC, and even SF9. All the presentations today will be covering CHO, and we specifically only use the CHO protocol. So you can be confident that this single CHO protocol is efficient and suitable for all CHO manufacturing cell lines. Now, even though we have a list of well-defined protocols, that doesn't mean that the process isn't customizable. In fact, in the next case study, we will illustrate how MaxCyte transfection can help users quickly identify optimized conditions by modifying both DNA concentration and post-electroporation seeding densities, and that this allows users to toggle their transfection efficiency and production titers. So for this first case study, researchers decided to work with a proprietary CHO-K1 derivative called Chosen. Now, I'm sure many of you are already familiar with Chosen, but probably for its more commonly used method for rapid generation of stable clones. Here, instead, researchers wanted to determine the optimal conditions for transient antibody workflow production. This optimization was achieved by testing 2 different plasmid concentrations and 2 different seeding densities. Looking at a visualization of this workflow, we can see that chosen cells were mixed with an antibody-expressing plasmid, and that DNA was at a concentration of either 200 micrograms per mil or 300 micrograms per mil. On the next slide, we'll indicate this with a lowercase or capital D. Cells were then rested post-electroporation at 37 degrees C for 35 minutes in a shake flask, and then resuspended in an off-the-shelf standard culture media to either 5 million or 10 million cells per mil. Again, on the next slide, this will be indicated with a lowercase s or a capital S. Cells were maintained in culture for 14 days and then analyzed for cell viability and antibody titer. Now, MaxCyte's nomenclature here is a little bit different than you might be familiar with. We're talking about 200 or 300 micrograms per mil, but because we're working with cells at a density of about 2 E8 cells per mil, this DNA concentration works out to be roughly around the range of 1 to 1.5 micrograms of plasmid DNA per million cells. So looking at the results of this workflow, we can see on the left that we start with a very high cell viability in all transfection conditions tested, and that after 48 hours, we have maintained that very high cell viability. Looking at the right, for antibody titer production over 14 days, and starting at the very bottom in dark blue, we can observe that the lowest DNA concentration combined with the lowest seeding density resulted in the lowest antibody titer. Moving up from there, either increasing the DNA concentration or increasing the post-electroporation seeding density resulted in an increased titer. And finally, the best and highest titer was achieved by using high DNA concentration and high post-electroporation seeding density. And in this situation, we were easily able to reach greater than 550 milligrams per liter by day 14. Now, importantly, here we're saying low seeding density and high seeding density, but I do want to highlight the fact that MaxCyte's transfection process is incredibly gentle. So even though we say low and high, our lowest density is actually quite high compared to other technologies. So to summarize this first case study, we illustrated how MaxCyte is a flexible system that can be used to dial in titer based on DNA concentration and post-transfection seeding density. This also allows for the rapid optimization of a transient antibody production in chosen cells. So moving on to our next case study, we wanted to share a story of high-yield transient protein production using CHO-S cells. And really, we wanted to illustrate how using MaxCyte for process can be used for process development as well as scale-up. So if we briefly take a step back, I wanted to talk about a common pain point that many in the protein production field encounter. So while many people want to use CHO cells because it does have FDA approval for the expression of monoclonal antibodies, often people will use HEX cells in early development stages for transient production and antibody production, largely because of its ease of use and cost. However, at some point, you will need to switch over to CHO cells, which will then require some level of revalidation. And in a worst case scenario, the CHO cell product differs from the HEX cell product due to post-translational modifications, meaning that some of your previous testing and possible candidates can actually fail to function as expected. So I ask you to imagine a scenario where instead of spending your time developing a HEC protocol and then having to go back and develop a CHO protocol, you were able to save your company both time and money by starting your development process in your preferred manufacturing cell line of CHO. And so that's exactly what we're hoping to illustrate with this data set. Researchers chose the CHO-S cell line to do their early process development. They then did 2 rounds of small-scale optimization, mainly focused on post-electroporation culture conditions, and then replicated this best condition isolated from the small scale without any modification to the electroporation process at large scale. So to sort of visualize that workflow, again, here we are taking CHO-S cells, resuspending them in MaxCyte's universal buffer at a density of 2 E8 cells per mil, electroporating them with our CHO energy protocol in OC400 processing assemblies. Cells are rested in a shake flask and then resuspended in media. Now recall I mentioned that there was 2 rounds of media development here. In the first round of process development, researchers wanted to evaluate the effects of sodium butyrate as well as temperature shifts to maximize productivity. Sodium butyrate is used as a histone deacetylase inhibitor and also increases transcription and impacts the rate of cell division. Temperature shifting also can allow cells to slow down and focus their energy on production rather than cell division. So looking at the specific productivity of these conditions, we can see that individually, holding the cells at 32 degrees C or increasing the productivity with sodium butyrate without temperature shift does increase the specific productivity compared to 37 degrees C. However, it's the combination of these 2 conditions that actually yielded the best productivity for these cells in this specific media. Now, importantly, MaxCyte does not have any restrictions on what type of media can be used after electroporation, which gives our clients the flexibility to evaluate a range of media and feed combinations to give them the best balance of not only productivity, but also cost. For example, in the next round of process development, researchers wanted to evaluate two different base media. Media 1 is the same media that we explored in the first experiment, where media 2 is a common alternative base media. And then for a third variation, researchers added an easily available off-the-shelf supplement for production. Now, looking at viability again, we can see immediately, following electroporation, cells had a very high viability regardless of the chosen base media. These cells are able to then maintain that high viability throughout the production, which means cells are able to produce as much protein as possible. That's played out here on the right by looking at the titers, where we can see already that media 2 is able to generate a much greater titer compared to media 1, and that this production is enhanced by this supplemental addition. So from this early process development, we can see that due to MaxCyte's ability to give high transfection efficiency and highly viable cells following electroporation, scientists aren't losing time trying to develop a HEC process and then develop a CHO process, and instead are able to run all their small-scale experiments in their desired CHO cell line, rapidly evaluate a range of conditions, and develop a robust process that is actually capable, as you can see in the purple line, of producing over 1.2 grams per liter by day 14. Again, all in small scale. Moving on to the scalable workflow, and I think this is really where MaxCyte shines, researchers took the conditions that they had already developed during those small-scale process development steps and performed an experiment in parallel with the large processing assembly called CL2 without making any modifications to the culture media, electroporation, or DNA concentration conditions established at the small scale. So here, cells were electroporated with 2 micrograms of plasmid per million cells, and then cultured at a seeding density of 6 million cells per mil, and in the small scale cultured at a 20 mil volume, where the large scale was a 2.8 liter volume. Looking at the titers reflected on the left, we can see that there is a minimal difference in productivity rates just looking at time between the small scale and large scale. Looking at overall yields, from the small scale experiment, again, this is an OC400, we were able to generate 21 milligrams of usable protein, where in the large scale, we were actually able to generate up to 3.5 grams of usable antibody. So clearly, we were able to optimize conditions that are scalable. And this is one of the key takeaways that I want to make sure that you take away from this first case study. With MaxCyte, you can easily perform your process development work at the small scale, quickly determine the high yielding conditions needed for transient workflows, and then our users are able to seamlessly scale up their production without any reoptimization, and importantly, no loss of time. And in this case, for CHO SLs, we were able to develop a robust process capable of producing over 1.2 grams per liter. Now, so far, we've focused our attention on these transient workflow that takes about 2 weeks, which is really great for producing a lot of material quickly for early candidate evaluation and some level of process development. However, taking things further down the pipeline, I wanted to discuss a few stable workflows and specifically bulk pool strategies. Now, for those of you that are unfamiliar with bulk pool workflows, briefly, cells are transfected and then cultured in some type of selection media that kills off the untransfected population. These surviving cells are then expanded and cryopreserved and then later used for some type of production. Now, one of these commonly used cell lines for this is actually DG44, which is what we will be covering in this next case study. So DG44 lacks the dihydrofolate reductase gene and therefore cannot grow in an unsupplemented media. Researchers therefore exploit this deficiency by transfecting in antibody vectors containing the DHFR gene, which means only the cells that have been successfully transfected will survive selection. In some instances, researchers will apply an additional selective pressure by treating with the drug methotrexate that further inhibits DHFR, thereby selecting only the highly transfected population of cells, which should also correspond to cells highly expressing your antibody of interest. So the goal of this case study was to develop a stable DG44 clone and to evaluate the different ways of doing bulk pool and mini pool workflows. Now, because of time limits, I don't have the ability to go into too many details about what the major differences in bulk pools and mini pools, but I want to encourage you to check out the MaxCite website, which actually has an Ask the Expert button, which quickly connects you with one of your local FASs, who I'm sure would be more than happy to walk you through this stable workflow process in highly great detail. But at a high level, what I really just want to draw your attention to is the difference in how these cells are cultured and how many cells are cultured after electroporation. So for bulk pools, cells are seeded after electroporation into a bulk media containing lacking an HT supplement. For this workflow, once the cells recover from the selection, they are then put into a production and their titer assay. So looking at that first step where cells are seeded in a bulk volume and put into selective media, we can observe an increase in starting viability before selection. We can observe that MaxCite starts with a high viability before selection and a high viability during selection. So this process proceeds quickly, with selection and expansion occurring within just 3 weeks. Now, I'd like you to consider if you were using a different transfection methodology that either meant you started with a lower viability or a lower transfection efficiency. This would mean you not only have unhealthy cells going into a stressful selection process, but you also have a recovery process that will take longer because there are fewer cells expressing your gene of interest. In the race to develop monoclonal antibodies, this time loss can be extremely costly. And so again, this is a critical benefit to MaxCite users because we're reducing that timeline for you by shortening the amount of time it takes for those cells to recover from stable selection. So looking at that selected bulk pool that has now been put into a production culture, we can see a high viability and robust cell growth with a titer exceeding 100 milligrams per liter by day 18. Now, importantly, it is important to keep in mind that with DG44, this is considered a hard to transfect cell line. And just to make things more exciting for this case study, we decided to transfect in a difficult to express protein. So 100 milligrams per liter is an impressive result at this stage. Still, this does also highlight the drawback of a bulk pool, where you are limited by a mixed population of cells, which means you have both high expressing cells with that titer being averaged out by low expressors, all competing for reagents. And this is why a lot of researchers choose to pursue mini pool workflows. After electroporation, only about 2,000 cells are seeded in this experiment per well of a 96 well plate for selection. Of those 2,000 cells, you will end up with small different combinations of populations, with some wells having low expressors, some having a mix of both small, low and high expressors, so similar to something you might see in the stable bulk pool. But then you'll also end up with a handful of scenarios where you have a well that is only a mix of high expressing cells. So stopping at this point to just talk about the mini pools before we take that workflow in further. For this mini pool experiment, 20 plates of 96 well plates were seeded and selected in unsupplemented media for 3 to 4 weeks. From this selection, 240 mini pools were assayed, and their wells were assayed for a titer. The 15 best titers were then scaled up again for a fed batch production, a more classic fed batch production. So looking at this fed batch, we can observe the growth rates of these mini pools and confirm that they maintain a high viability throughout this production. Jumping ahead to look at the yields, we can see that these selected mini pools, some of them still have that mixed population producing a similar titer to the bulk pool, as you can see with the orange data set, or the orange mini pool, which is on the bottom. However, you can also easily identify that pool 140 has several highly expressing cells and is able to reach a titer nearly twice that of the bulk pool. So briefly, where we can go beyond this is to take that selected mini pool, number 140, and further amplify this with a round of methotrexate drug selection. And then additionally, a further evaluation could be to take that and do single cell cloning. So looking at all 4 variations of this workflow, on the left we have the bulk pool, then in purple we have the original 100, the mini pool number 140. Then we have that amplified mini pool. So this is that mini pool that has undergone methotrexate selection. And then finally on the far right, we have the stable clone version that took about 3 months to generate. So I think that this is a really impressive illustration of how mini pools can take your process forward and give you a high yielding stable clone within 3 months. So from this case study, we can observe that there are many benefits to pursuing mini pools to identify a high yielding clone. Additionally, due to the high transfection efficiency and cell viability post-electroporation, fewer plates were actually needed to be screened to identify this high yielding mini pool compared to other technologies, and that you can take this forward to cell line development. In fact, MaxCyte really does provide you with the flexibility to explore the workflow that works best for your timelines. So even if mini pool is not the strategy you want to go forward with, you can still see how well the bulk pool process also works. So as I've mentioned, we've now covered small-scale transient workflows. We've also covered a bit of stable pools and stable cell line development. But to walk us through the process of rapid stable cell line development, I'd like to pass things over to Jim Brady. Take it away, Jim.
Thanks, Megan. So just to highlight what Megan has showed you so far, we've discussed how MaxCyte provides multiple advantages for rapid transient protein expression and for generation of bulk pools and mini pools. Now, as therapeutic developers move closer to the clinic, larger amounts of protein are required for in vivo studies and then ultimately for clinical testing. The demand for larger protein quantities is typically met through the generation of clonal cell lines. But today I'd like to take a few minutes to talk about how high efficiency and viability of MaxCyte's transfection process can enable more efficient workflows for stable cell line generation. Now, the next couple of slides provide an overview of the workflow for generating the stable cell line by limited dilution cloning following electroporation of CHO-S cells with a bicistronic antibody plasmid that contains a G418 selection cassette. I'll also share data on product quality and clone stability. Now, in this example, the cell line development process began with generation of a stable bulk pool by applying G418 selection 1 day post-electroporation. As Megan noted earlier, MaxCyte transfected cells typically recover from selection more quickly versus cells transfected with other methods. In this case, a stable pool was generated within 2 weeks after electroporation. Clonal cell lines were then produced by limited dilution cloning, by plating cells at a density of 0.3 cells per well and 96 well plates, followed by ELISA screen to identify high titer clones. I'd like to highlight that only 25 plates and 479 clones were screened in this study, and the work was accomplished by a single scientist. Normally, much larger numbers of plates and clones would be needed, and we would have to use expensive automated clone picking technologies to find that rare high titer clone. And then finally, the top 23 clones were expanded in fed batch cultures using commercially available media and feed supplements. And note that this manual process took less than 8 weeks from electroporation to final titer analysis. Here are the antibody production kinetics from the top producing clone, which yielded over 3 grams per liter in an unoptimized fed batch culture. And then after further process development, the same clone produced a volumetric titer of more than 5.5 grams per liter. Now, in addition to seeking high titers, cell line developers want to verify that titers and critical quality attributes are maintained after serial passaging. To generate stability data, we established a cell bank right after initial clone expansion, and then we created 2 more banks after expansion for an additional 30 and 60 days, respectively. Clones from all 3 banks were thawed at the same time, expanded, and then seeded into parallel production cultures. You can see post-translational modification patterns assessed by glycoform analysis were consistent across all 3 banks of cells. And after 60 days of serial passaging, titers dropped by less than 15%, demonstrating that this clone was highly stable. So in summary, a high titer clonal cell line was generated within 6 weeks of electroporation, and clonal stability was demonstrated for at least 60 days. This case study illustrates how MaxCyte provides multiple time and cost savings for stable cell line development, including lower consumable costs, fewer FTE hours, and elimination of the need for expensive clone picking instrumentation. I'd also like to highlight that MaxCyte allows therapeutic protein developers to parallel track stable cell line development while generating data with protein produced transiently following large-scale flow electroporation. In this sample workflow, 20 billion CHO cells were transfected with an antibody plasmid using the CL2 processing assembly, and then immediately after electroporation, 80 million cells are subjected to a selection for stable pull and clone generation, while the remaining cells are inoculated into fed batch culture to produce protein for CMC studies. If the candidate fails to meet acceptance criteria based on analysis of transiently produced protein, the developer can terminate the cell line development process, which results in critical time and cost savings. Now, of course, in order for transiently produced protein to serve as a surrogate for protein from a stable cell line, it's essential for the transiently material and the stably produced proteins to have consistent critical quality attributes. 2 of the most important contributors to protein quality are the host cell and the composition of the culture medium. As Megan mentioned earlier, the key advantage of MaxCyte's technology is that it offers the flexibility to work with any host cell line and media formulation. Here we perform glycan profiling and gel analysis of an antibody that was expressed via 4 independent transient transfections, and we compare those proteins to the same antibody expressed from a clonally derived cell line. The same host cell line, basal media, and feed supplements were used in all 5 production cultures. And you can see identical banding patterns across the gel, indicating that none of the cultures produced degraded or aggregated proteins. And on the right, you can see glycosylation profiles are also consistent, indicating reproducibility of the transient production process and demonstrating that proteins expressed via transient and stable production following MaxCyte electroporation are highly consistent in quality. So in summary, we've shown that MaxCyte enables high titer transient protein production in less than 2 weeks, and the quality of the protein produced by MaxCyte transient transfection is consistent with protein produced from a stable cell line. MaxCyte provides significant time and cost savings through the use of preferred manufacturing cell lines during early phases of discovery and development, and by delaying the requirement for stable cell line production. As we mentioned earlier, the MaxCyte STX instrument can transfect up to 20 billion CHO cells at one time, and that number of cells will typically yield hundreds of milligrams up to a few grams of a conventional antibody, which can address many early to mid-phase discovery and development needs. However, after talking to industry experts, we realized that a larger scale of transfection is required to produce the quantities of protein needed for preclinical testing and in vivo studies. And that's why we developed our Expert VLX instrument. Now, this is the workflow that we just discussed for parallel tracking large-scale transient protein expression with stable cell line generation. And with the STX, it's possible to generate low-gram quantities of transiently produced protein within a couple of weeks. And this parallel workflow is a great way to save time during the development process and reduce the risk of investing in stable cell line development for projects that could fail at the CMC stage. But what if you could extend this concept even further and use transient material to do more? Now, the MaxCyte VLX instrument goes one step further, transfecting up to 200 billion cells in one process, potentially generating tens of grams of protein in that same 2-week time frame. In order to do this, the VLX needs to integrate into a closed process workflow, so that's what we're going to look at next. Now, before discussing the VLX workflow, I'd just like to highlight some of the key features of the VLX instrument and consumables. This is a larger view of the VLX instrument, which is able to efficiently transfect up to 1 liter of concentrated cells in under 30 minutes. As with all of our expert instruments, the VLX is controlled by a touchscreen interface, and pre-optimized protocols are selected from a drop-down menu. Here, the instrument is shown with a single-use 1 liter flow processing assembly attached to the instrument. Based on the same principles as our STX flow electroporation process, sterile air is used to gently move cells in and out of the electroporation chamber. The electroporation chamber is larger on the VLX, so 1 liter of cells can be processed in less than 30 minutes, which is comparable to the maximum processing time on the STX instrument. Also, smaller static processing assemblies can be used on the VLX to process control samples. Here's a larger view of the R1L processing assembly. The sample and collection bags are connected to the electroporation chamber with bioweldable tubing that conforms to bioprocessing industry standards, allowing close connectivity with upstream cell concentration and buffer exchange technologies, as well as connection to downstream cell bags and bioreactors. Ports with wide connectors on the sample bag allow for serial introduction of loading agents and other reagents prior to electroporation. This gives an overview of the steps involved in large-scale transient bioproduction with VLX electroporation. Because of the large number of cells we'll be electroporating, the pre- and post-electroporation steps are critical. For the remainder of my presentation, I'll describe a case study based on developing a process for transient antibody production in CHO cells with the VLX. Now, multiple different bioreactors and various methods of cell concentration and buffer exchange can be used in a closed system with the VLX. In this case study, we cultured CHO cells, in this case chosen cells specifically, in a BioFlow 320 bioreactor for seed train expansion prior to electroporation. Then for downstream cell culture, we also used a BioFlow 320 reactor for antibody production. The Sartorius KSEP 400 was used for cell concentration and buffer exchange upstream of the VLX, and the cell bag was used for cell resting immediately after electroporation. Focusing on the pre-electroporation steps, cells were expanded in the seed train bioreactor to a density of 10 million cells per mil. And in this particular study, 5 liters of cells were transferred to the KSEP 400, where spent media was removed and replaced with MaxCyte electroporation buffer. In this particular study, cells were concentrated to a density of 100 million cells per mil. The final recovery was 93%, and cell viability was 95% after cell concentration and buffer exchange. Antibody plasmid DNA was then added to 400 mils of concentrated cells in the R1L processing assembly. In this workflow, we removed the collection bag from the R1L, and we sterilely welded the output tubing of the R1L to the input tubing of a cell bag. So the cell bag provides a larger surface area, which facilitates cell recovery immediately after electroporation. After a 30-minute resting step, media was added to the cell bag, and the electroporated cells were then transferred via gravity flow into a downstream bioreactor for production. Cell viability, viable cell density, and titers were measured in the large-scale electroporation cultured in the bioreactor, as well as in parallel shake flask cultures containing cells that were transfected in the small-scale static processing assemblies. We also established 2 satellite cultures in shake flasks with cells taken from the large-scale VLX flow electroporation. You can see high starting viabilities immediately post-electroporation, the graph on the right, and viabilities remained above 50% for 14 days in all cultures established from the large-scale electroporation. Antibody titers reached 700 mg per liter in the bioreactor and into the satellite cultures established from the large-scale electroporation. The graph on the right shows consistent specific productivities across all cultures, including the 1 ml static control, thus demonstrating scalability of MaxCyte's electroporation process. So, summarizing some key features and benefits of the Expert VLX instrument, up to 200 billion cells can be transfected in less than 30 minutes. The technology can easily be integrated into a closed process. Transfected cells exhibit high viability and desired growth kinetics. And finally, we demonstrated that the VLX enables large-scale transient bioproduction in chosen cells for GLP and cGMP manufacturing, providing faster lead progression from concept to clinic. So, in conclusion, MaxCyte's electroporation process offers flexibility to meet your specific project goals and development timelines by providing rapid, stable cell line generation, high transient protein expression yields, and parallel workflows for accelerated therapeutic protein development. Before we conclude, I'd just like to mention that we will be at Bioprocessing International in Boston next week, where MaxCyte's Senior Director of Process Development and Innovation, Joseph Abad, will be speaking on Wednesday. We encourage everyone to stop by our booth and to attend our sponsored networking event at Spin Boston on Tuesday. So at this point, I'd like to thank everyone for listening, and we're happy to answer any questions from our audience.
Thank you, Caroline, Megan, and Jim, for your informative presentations. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located in the lower left corner of your screen, and we'll answer as many of your questions as we have time for today. Let's go ahead and get started. Our first question here is, can you provide more details on how you scale up transfection using MaxCyte electroporation?
Yeah, I can take that question. So, as Megan highlighted earlier, we have pre-optimized protocols for a range of cell types, including CHO cells. So at any scale, you don't have to try to identify the right parameters such as voltage, pulsing, et cetera. You just select the CHO protocol from a drop-down menu, as Megan also showed. At that smaller scale, you can optimize other variables like the DNA concentration, the cell seeding density post-transfection, the media feed strategy. But once you've locked in those critical process parameters and you want to scale up the process, you just concentrate cells to that same cell density used at small scale, add the same concentration of DNA, and then you can just transfer those cells to a larger processing assembly. We have processing assemblies that cover a range from 15 microliters to 1 liter of concentrated cells. So you could do anywhere from a few thousand cells to hundreds of billion cells in one electroporation. But the scale up just involves putting the cells into a larger chamber and then selecting that same protocol from the drop-down menu.
Wonderful. Thank you so much. Our next question here that we have, how does MaxCyte's electroporation-based transfection process compare to chemical-based transfection methods such as lipids or PEI for recombinant protein production?
Okay, Megan, did you want me to take that one as well?
Yeah, I can address that.
Okay,
so,
go ahead.
Yeah, so electroporation is definitely a different transfection method compared to those lipid-based methods. But because transfection creates pores in the cells and gets the cargo into the nucleus very, very quickly, one of the first ways that MaxCyte electroporation is different from these chemical and lipid-based methods is how quickly the cells can begin translating that DNA and start making that protein. So in that way, I would say MaxCyte is a faster process. Additionally, because there is no batch-to-batch variability, either between the manufacturers or there's no batch-to-batch variability with MaxCyte. So in a PEI or lipid, you're either basing that on the quality of product being manufactured by that company or your scientist's ability to create that lipid-based particle. So that's another great example of how MaxCyte is a better process, in my opinion, compared to those other technologies. Jim, is there more that you'd like to add there?
Yeah, those are great points, Megan. I just had a couple of other points. People have been successful at using PEI or lipids to transfect CHO cells, but CHO cells in general are challenging to transfect, and when people have been successful, they've either had to work with an optimized CHO cell line that responds well to those reagents, or they've had to use special media feed strategies to work with reagents. And so, some of the key advantages of MaxCyte is that we can work with any CHO cell line and any base media, and you don't have to find a CHO cell line that meets your transfection method. Rather, we have a transfection method that works with your cells and your media.
Excellent. Thank you both for that. Looks like we have one more question in here. Can you elaborate on critical post-electroporation process development parameters for maximizing transient protein titers?
Megan, you want to take that one as well?
I can take a little bit of it. I think I talked a lot about that at the beginning of my talk. So along with choosing an appropriate media feed strategy that works well for you, it's also important to consider the seeding density post-electroporation. Another critical parameter, while it doesn't necessarily happen post-electroporation, is really that cell viability going into electroporation. So MaxCyte is a very gentle process, but if the cells are unhealthy going into the process, they're not going to be healthy coming out of the process. And that's going to result in a lower starting viability, which means you've got fewer cells producing the product that you're hoping to manufacture. Additionally, as a MaxCyte FAS, one of the things that I like to promote is that you have access to the FAS team. So we can help you identify and develop those parameters to test and review, as well as make educated suggestions on materials to test. I'd like to throw that to Jim to see what his answer might be on that as well.
Yeah, and just to kind of elaborate a little bit on what Megan just said, one area where transient protein expression is different from making protein from a stable cell line is that with a stable cell line, you typically seed the cells at low density and then expand them in a bioreactor. Whereas with transient expression, we recommend starting at high density, and then you want to keep the cells at that high density so they hang on to the DNA and produce protein as quickly as possible. And of course, when cells are at high density, we have to keep them happy and well fed. And that's where process development comes into the picture. And, we have an array of customers. Some of them have very sophisticated approaches to process development, where they monitor metabolites and glutamine and glucose and dissolved oxygen. And then they can adjust their feed strategy based on those readouts. And then we have other customers that want a very simple, robust process where they can put cells into a flask or bioreactor, add feed a couple of times, and then just let the cells go. And we have recommended protocols for both sets of customers. And as Megan pointed out, we have an internal team of application scientists and the field team, and we can work with you to develop a production process that's right for your proteins and your skill levels.
Excellent. Thank you again, Jim, Megan, for your time today and your important research. We would also like to thank LabRoots and our sponsor, MaxCyte, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions that we did not have time for today and any questions submitted during our on-demand period will be addressed by our speakers via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, thank you and take care, everyone. Goodbye.